to present joint work of my group together with the ETH Zurich, the proteomics group, Roche AG, and Gene Data AG in Basel, and toxicologist from Maastricht University. So what I would like to show you today is that we can, if we interpret omics data, multiple omics data on the network level, that we can derive a nice kind of solution for data integration and derive biomarkers from that. And that comes from the following observation. So typically, if you make a proteomics experiment and a transcriptomics experiment on the same, let's say, cell line or patient, what you usually observe is you get a list of differentially expressed protein, a list of differentially expressed genes. If you then overlay this list, the actual intersection is quite low. However, if you map this list of proteins to pathways and this list of genes to pathways, at the pathway level, you see that both omics results basically enrich the same pathways, as you see in this example. So that led us to the idea to basically use the network as a scaffold for data integration. This is also important for precision medicine, where you typically draw multiple omics data from certain patients, and you try to find, let's say, thematic networks, hotspots in the networks that may be targeted by drugs. And people have shown that network propagation actually is a way of integrating multiple data types. They are able to boost the signal-to-noise ratio of a single protein and genes. Important for proteomics, for example, is that you can handle incomplete information, because in proteomics, typically you observe hundreds to thousands, a couple of thousands of proteins, but not the full proteome. So you have many proteins that you cannot identify. And what you get also is a functional interpretation of your complex data sets. Now, in this talk, what I want to show you is a way, so let's say a schedule for network-based analysis of multiple omics data. And I want to show you our application, which is a case of drug toxicity. In that case, anthracycline cardiotoxicity. What are anthracyclines? So anthracyclines are heavily used in clinical practice. They have been found, they are antibiotics. They have been found years ago. The first one was daunorubicin in 1963. Then we have doxorubicin, idarubicin, and epirubicin. They have more or less a similar mechanism. They intercalate with topoisomerase and interfere cell division of cancer cells, but unfortunately also normal cells. And they are among the most effective anti-cancer drugs to date. They are heavily used in breast cancer. However, they have side effects. They lead to short and long-term cardiotoxicity, depending on the cohort, in 5 to 23% of the patients, which is really a severe problem. Women got cardiotoxicity problems even years after therapy, so it's really a long-term effect. And on the molecular level, ACs induce multiple forms of cellular injury to cardiomyocytes. Most importantly, they are metabolized and lead to ROS production, which then damage the mitochondrion, so interfere with ATP production, energy generation for the cell. They inhibit DNA and RNA synthesis in the initiation and elongation phase. They intercalate with topoisomerase, and also which means the RNA synthesis, DNA synthesis is interfered. And there have been found a couple of biomarkers, yet most importantly troponins, or also some microRNAs. But what we can state from the state of the art actually is that AC-induced cardiotoxicity is really multifactorial and highly complex. Now our system where we want to do the analysis on is an in vitro system. We cannot do it in patients, of course, but it's a special in vitro system. It's not normal cell lines, but it's 3D microtissues. And that is cool because these 3D microtissues, basically it's a mixture of cardiomyocytes and fibroblasts at a ratio of 4:1. They consist of around 5,000 cells, and they have stable physiological characteristics over a long period, which is essential. Those characteristics, for example, are contractility. You can look at under the microscope, they really contract. It's fascinating. They remain the size, and also the ATP production, which is important for muscle contraction, is preserved. So we took this system, and then we treat the system with four anthracycline, DOX, EPI, IDA, and DAO. And we did a long time series over 14 days at 7 different time points. And we did transcriptome measurements and proteome measurements, always compared at each time point against the control. And we did 3 replicates for treatment and control. So what we could in principle do is for each time point, look at the differences in transcriptome and proteome, generate a lot of lists, and then scroll through that list, through these lists, and try to find interesting markers. What we instead were interested in was to really capture the dynamic changes induced by the anthracyclines. So we want to keep the entire time series and got protein candidates from the comparison of the treatment time series against the control time series. And of course, we want to integrate the data at the beginning of the analysis and not at the end of analysis by comparing lists, but start with the integration at the beginning. And of course, we were interested in drug response networks and biomarkers of cardiotoxicity in vitro. And then we want to extrapolate that, of course, to the clinical case. So the tool that I want to describe is called network propagation. That's basically an agglomeration of tools. What does it do? It's all guided by the guild by association principle. That means network propagation on protein-protein interaction networks. So the guild by association says if 2 proteins interact, they are somehow functionally related. Yeah? So the interaction of the proteins tell us something about the similar function. So once you have your protein-protein interaction network and you have your initial experiments, that is the weights of the nodes. The nodes are the proteins. The interactions are physical interactions between proteins. And then network propagation is nothing than a stochastic process that traces the flow of information through the network over time. And the most important thing is probably random walk with restart, which is very well known. So suppose you have a protein expression in 2 of the proteins, A and H in that case, then of course, not only these proteins are affected by your treatment, but also the proteins that are interacting with these proteins. For example, D in that case. So what you do is you take the initial weights and spread them to the neighborhood. In that case, this gene has only this gene as a neighborhood, and this gene has these 2 genes as a neighborhood. So the neighborhood gets also weight from the initial measurements. Then you track that until you reach a certain convergence stage or steady state. And at the end, you have basically reweighted your network by the information flow from the initial experiments. Then you cut and test you cut basically the nodes from the tree that do not have enough weight. Enough is always a statistical significance test. And then you've tried to find connected components. And these are basically your final network modules that you are mostly interested with. Now, this convergence has a nice form to compute it mathematically. So if Zk is the vector of node weights at time point k, it's a sum of the initial node weight vector Z0 and a multiplication of a matrix W and the weight vector at time point k minus 1. And this converges to this state. And just to highlight it, this convergence is dependent only on the initial vector of nodes that we have, which can be computed or which you have already, and basically a matrix W that describes the graph, the interaction graph. So both components are very important, the interaction graph that you use and of course, your experimental data. So coming to the interaction graph, in my group, we developed the consensus PathDB as a resource for human interactions. There are a lot of resources out there. The CPDB is one of them. What we basically did is we integrated interactions from 33 different public resources. Most well-known are REACTOME, KEC, and other resources. We have different types of interactions like physical interactions, gene regulatory interactions, drug-target interactions brought together into a big graph. For this study, we use only the part which is constructed from protein-protein interactions. And we use that as an initial scaffold that gives us our protein interaction network. Then we score, we need the scoring. So the initial vector Z0. This is done basically by a polynomial regression model, taking the 7 time points as an entire time series, compare this time series or fit a polynomial to the treatment time series and the control time series. And when this is different, we give a high significance p-value to that particular protein or to that particular gene. We performed a total of 200 LCMS experiments and 200 transcriptome RNA-seq experiments. And by that, we identified around 641 proteins, which are dynamically changing, and 900 different genes, which are dynamically changing. And what we do is then we initialize our protein-protein interaction by the p-value that we get for each protein and each gene if this p-value is significant. And you see that this is a very nice data integration concept actually, because if you have transcriptome plus protein, you simply add the 2 p-values. So what we have at the initial state is we compute for each anthracycline 3 networks, one derived from the transcriptome data, one derived from the protein data, and one derived from the combined data set. Now, this is the approach so far. We have our protein-protein interaction network, we have our scoring system, and we do the network propagation. And then we have these 3 initial networks for proteomics, transcriptomics, and combined. And what we compute from these are 3 different network modules, which claim to agglomerate the hotspots of the data. Now, what we see when we have these, for each anthracycline, we have these 3 networks, what we can ask at the end, what is the functional enrichment gained by the different data sets? And these are the different geo categories. And what you can see is that if you sum them all up, it's basically that the integrated approach has the highest functional enrichment, followed by the protein, followed by the transcript. So even if you have less proteins, you had 600 or so proteins, 900 or so transcripts, protein is still more functionally relevant than transcripts. Now, what we have is basically for docs, epi, ida, and DAO, we take now the integrated network, but we have still 4 networks, and we integrate them. So we integrate them to what we call AC response network. And what you nicely see is that response network follows in 3 major compartments: the sarcomere, it's the contracting muscle, the mitochondrion, and then the extracellular matrix, which very nicely reflects literature. I just hopped through the results because of time. We did a phosphorylation experiment to see whether our response network has a lot of signaling activity, and indeed it has. So many of the proteins, significantly more than you would expect it, are phosphorylated. We also looked at clinical impact, and we found that we can we find biomarkers in this network that are expressed in human biopsies, and that also correlate with the clinical parameter, the left ventricular ejection fraction. So we got nice biomarkers out of that. We also detected missing information. For example, we detected a transcription factor which hasn't been identified by proteomics. It was only partially identified at several time points, but not an entire time series. But it came into this network because it had many relations to sarcomere proteins. And yeah, that is just to summarize. Sorry for hopping through the last slides, but you can ask me in the break. So we think that a network approach can integrate different omics data sets, and it's based on information propagation through PPI networks. We have constructed a high quality PPI interaction network with our tool consensus PathDB. We applied a dynamic scoring approach to integrate both LCMS and transcriptome data. We saw that the biological function enrichment is highest in the combined, followed by protein, followed by transcript data. We combined all anthracycline network modules to a response network, and we saw that many of them are phosphorylated and also expressed in biopsies, and we derived biomarkers from that. They also have a direct impact on mitochondrial function, which I skipped here. And this approach also is able to identify missing data, for example, proteins that haven't been measured, but that come with a network because they have interactions to other genes. These are the acknowledgments, the project partners, and the funding organization. And thank you for your attention.